Hi, I'm Jim Paterniti. What you're seeing on your screen now is a picture, an electron micrograph actually, of a COVID-19 virus, which is the SARS-CoV-2 virus. It's responsible, even though it's pretty, for the death of worldwide almost 450,000 people, probably by the end of the time. It's time, it'll be more than that. Right now, we're coming out of lockdown and we're reopening. And what you're looking at are a group of graphs of different states, of the number of cases by day in the different states. And although California isn't listed here, it's right on the same pattern. And this is exactly what we would expect because the virus hasn't changed. What's changed is our behavior. Today, though, we're going to talk about how we can, or what we can do to protect ourselves ultimately, and what other people are doing to help society to overcome this scourge. We're going to look at the measures to produce vaccines, get a picture of the real-world timelines for getting them to people, and to instilling herd immunity. And we'll explain what that is. And lastly, as I say, we'll talk about the effect of the pandemic on people with Parkinson's. So that's what we're going to do, and there'll be a Q&A probably at some point. In order to protect us from pathogens, we've evolved an immune system, which we're going to talk about in some detail. Before we do that, let's define some terms. First of all, what is a pathogen? A pathogen is a disease-causing organism or a toxin. Examples of the disease-causing organisms would be the viruses. There are also bacteria. There are parasites, there are fungi, a whole host of coal tar, a whole host of agents that seek to hurt us. And anything that triggers the immune response is called an antigen. So when you hear the term antigen, that's just something that causes the human body to react and jazz up its immune system. An epitope is the part of the antigen that is recognized by the immune response. So if you have a big ball, like that COVID SARS-2 virus, it's just those little crowns that probably are going to be antigenic. That is to say, they'll be recognized by the immune system and hopefully we'll make antibodies against them, but we'll talk about that in more detail. The other words you should know is a prion, which is a small misfolded piece of protein, which we can discuss in connection with mad cow's disease, but also with respect to Parkinson's. It has some interesting aspects. And finally, if I use the term nucleic acids, I'm referring to DNA in all the forms of RNA. And this is the genetic material that determines how cells function and reproduce. And when a pathogen, so that's the disease-causing organism, challenges us, we can ward it off with a variety of different tools that we use. Some are innate to us, and that forms the components of the innate immune system, and some are adaptive responses. That's part of our adaptive immune system. So I'll go through some of the highlights of how each of these systems works in concert with the other parts of the immune system, and it helps us to overcome pathogenesis, the disease-causing process of disease. Okay, first one is really cute. Situational awareness. So really behavior is part of your immune system. In the act of wearing a mask, the act of social distancing, the act of cleaning your cutting board after you're done cutting up the chicken, all of these behaviors that we have, brushing your teeth to make sure that they don't become infected. All of these things that we consciously do with our brains are part of our innate immune system. And we're the only animals that are capable of doing this to the degree that it's being done. I mean, nobody else builds hospitals, nobody else makes masks. And so just think about that in terms of Parkinson's, when sometimes we lose a little bit of our behavioral awareness. We lose a little bit of our situational awareness. That's the time when we can make mistakes. And that's the time we need to be more vigilant. The next part of the innate immune system is called the immune surveillance system. This is kind of fun. Most of you know that there's another whole circulatory system beside the blood circulatory system in the body, and it's called the lymphatic system. And it consists of canals that run in very many cases parallel to the veins and arteries. And it's there to drain excess fluid from the body, but it's also the place that the surveillance for immune function in the innate system lives. So basically, the little nodes in your neck that swell up when you get sick sometimes, those are the lymph nodes that are part of the immune system. And they swell up because the cells in there, which are white blood cells of various types, are busy tackling the pathogen, in some cases digesting it, in some cases trying to inactivate it, in some cases breaking off pieces of it to present to other cells so that they can make antibodies. And so we've got this whole second circulatory system that is big, extensive, and provides us with this level of innate immunity. While we're on the subject, the largest organ in your body is your skin, and it's part of the innate immune system. Unless you can breach the skin, you can't get into the body. If you don't have a cut or a wound, if your skin is perfectly okay, you can ward off a lot of insults and a lot of attempts at breach. So, and then think about it. You then have mucous membranes in your mouth. They're a little bit more porous, and in the back of your throat and your lungs, and that's where you get your head colds. That's the reason the virus can only enter through the mouth of the nose. That's the reason we're wearing the masks for the coronavirus, because the mucous membranes are susceptible to breaching by the viruses. But in addition, we have saliva, and believe it or not, saliva's got enzymes in it, and it clears the mouth of whatever's there, and once it goes down your esophagus, it goes into your stomach, which has 1 molar hydrochloric acid. Anybody who was awake during chemistry class in high school knows that 1 molar hydrochloric acid will eat brick. And actually the only thing that protects us from having our stomachs eaten out is the mucus that we make that lines the inside of our stomach. But that's a perfect trap for bacteria or for viruses. And many infections are warded off in that way. In addition to your skin and your mucous membranes and your saliva, there are good bacteria that colonize your intestinal tract, your GI tract. And these guys bind to little receptors in the walls of the cells, the same way that COVID virus binds to the ACE1 receptor in the lungs. These good bacteria bind to their cognate receptors in the lining of the gut, and they prevent anything else from binding. And so if you have a UTI, or if you have a gut problem, they give you probiotics to restore the flora, to restore these good bacteria to colonize your gut and protect you against the pathogens that can cause the disease. The other thing we do that is very hardwired, actually, and part of the reptilian brain, it's the reflexes controlled back here, is vomiting. Vomiting is probably the single hardest wired reflex we have. If you couldn't vomit, you would be in tremendous trouble. So those are the components of the innate immune system, and they work. The key to the innate immune system is that it is triggered early, within minutes to hours, and it is very, very broad. It has very broad specificity. Picture the amount of protection from the different things that you get from just the skin alone, the different kinds of organisms you can protect against. In addition to the innate immune system, we have the adaptive immune system. And this is only found in vertebrates. This is only found in us and fish and birds and reptiles. And it's not found in insects. And the reason why is that you have to have a central nervous system in order to have an adaptive immune response, because your brain actually is important in controlling these things. The bioactive molecules that are made and released by the brain control aspects of the immune system. And if you don't have that in place, you can't have an adaptive immune system. The other organs that are involved in the adaptive immune system are your thymus, which is this lump of flesh underneath your breastbone, just in the front of your chest. And it makes lymphocytes, so-called T lymphocytes. So if you ever hear that term used, they process and display antigens so that the killer T cells, which are also made in the thymus, can go out and engulf if they're bacteria or virus particles, and clear them from the system. The other components of the adaptive immune system are the bone marrow, or the myeloid, if you ever hear that, or the B cells. And B cells are the ones that make the antibodies. And they also have one other function that's critical. They have what are called memory cells. The memory cell's job is to store the information about how to make a successful countermeasure against the bacteria or fungi or whatever the pathogen is. Even years down the pike, you know that your smallpox vaccination is good for life. Ideally, that's the way a vaccine should work. It creates enough memory cells which live long enough to provide lifelong protection for you. One of the critical issues of influenza, for example, the reason why you have to get an immunization every year is that the virus mutates. The memory cells can't find the virus because it's the old version of the virus, and you have to start from scratch. And so mutability of the virus is one of the ways that you can be foiled in terms of trying to make a vaccine that works for everything for a long time. The other things, so the aspects of the adaptive immune system that are important are that it works slower. It works in the order of days, weeks, but it's incredibly specific. I mean, antibodies recognize specific epitopes on the antigens, which are the pathogenic bacteria and viruses and fungi and prions and sometimes parasites. It's one B cell, one antigen. Every B cell that is differentiated is specific for an antigen, and it's thought that there are 10 million different antigens that are currently cataloged in your body, which is a pretty impressive number. Well, hasn't been lost on anyone that this information that I just gave you has implications for public health. And scientists learn to take advantage of the adaptive immune system to make vaccines, which activate antibody production against specific antigens. And it works. The smallpox vaccine has eliminated smallpox. 40 years ago, the last case of smallpox was recorded. There hasn't been a case since. It doesn't exist anymore, except in laboratory, in some cold room, ice box in Russia or in the US, behind double locked doors, hopefully. Get rid of that eventually. The next one due to become extinct is polio, and that's through the work of the World Health Organization and the Gates Foundations and the non-governmental agencies and so on. The only place you can find polio is about 500 cases in South Africa. And I think they're going after this year and next year, and the goal is to have a polio-free world within that period of time. Actually, medical sciences produce vaccines to about 30 diseases. Of course, it isn't all wine and roses. Vaccines normally take years to develop because of something called the inverse ratio of risk and benefit and the golden triangle. And I'm going to explain those two terms because these are two of my favorite concepts. And they're tremendously important in understanding why it takes so long to make a vaccine and why we have to calculate our timetables a little bit different from the way that the public officials have been calculating their timetable. Risk-benefit first. Let's talk about that. Keeping in mind the idea of doing no wrong, in contrast to treating a disease and taking some risk, vaccines are given to healthy people, and they're given to healthy people by the millions to avoid a disease. So it's not going to make you better, it's going to keep you from getting sick. So that's a probability, as opposed to a cancer drug that you take knowing that it's going to make you vomit and then it's going to make you feel like you have the flu and that you're nauseous. And in some cases that you're going to hold another 10 pounds of water. All of those side effects you withstand because you know that ultimately the drug is going to be killing the cancer cells faster than it's going to be killing you. And so you accept that risk-benefit ratio. With the vaccine, it hasn't happened. You're trying to prevent it from happening. And that means that has to be very, very safe because you're going to give it to millions of people. And we'll explain why you have to give it to millions of people when we talk about developing herd immunity. But this risk-benefit is inverse risk-benefit ratio is clearly one important aspect that you have to think about when you develop a vaccine. This is the golden triangle. The sides are good on the bottom, fast on the left side, and cheap on the right side. And this is actually interesting because the approach that the federal government is using to developing a SARS-CoV-2 vaccine is to go fast and good, hopefully. And that's the thing, it's not going to be cheap. They're going to be proceeding at risk, but they're going to try to move as fast as possible. The risk here is that they're going to build factories, manufacture doses of viral immune vaccines before they have the clinical data that says that things are safe and effective. So that's an at-risk project. And the other thing that the world is doing is that there are probably 130 or 140 different labs all working on vaccines to the COVID virus, SARS virus. So, for example, Moderna in Massachusetts is an early leader, and they're actually conducting a collaborative program with the National Institute of Allergy and Infectious Diseases in kidney. That's where Tony Fauci is the boss. And they are using an accelerated timeline that brings a drug from discovery through to marketing in about a year and a half, he says. And the way that they've done it is by silly wild-ass guessing. They're tremendously leveraged in terms of risks. They've also streamlined the clinical development. So in phase 1, when you take a drug into the clinic, and I've taken about 8 into the clinic, and I've written 2 INDs, so I can tell you from experience. The first thing you do is you take healthy volunteers and you determine whether or not the vaccine is safe in that small population. And roughly, if you can get any indications of efficacy, that's great. If not, you try to push all the safety features that you can push. The second thing you do is then go to phase 2. And in phase 2, which usually takes 6 to 11 or 12 months, in contrast to phase 1, which takes about 3 months. In phase 2, you have a larger number of people. Usually maybe there'll be 45 in the first phase and 200 in the second. Phase 2, you set the doses, so you give 10 milligrams or 10 micrograms and 100 micrograms, 1,000 micrograms, or however you want to grade the doses. And you look this time for little signs of efficacy if you can find them, but definitely for the maximum tolerated dose. Because if you don't have efficacy at the maximum tolerated dose, you're done. You don't go any further. Once that's established, that information is used to power phase 3. Phase 3 is your golden phase, where you do, in general, 2 clinical trials of 1,000 or more people, and you demonstrate safety and efficacy. In addition, in groups like the elderly, people with renal impairment, pregnant women, special cases like that. And then after that, you put all the data together, you submit an NDA, and you send it in. If it's approved, you then go and market your drug, but you're still bound to do after-market surveillance, which is called phase 4. And that's when doctors and patients are monitoring. You get information that allows you to get back to the government with specific adverse effects. And the government keeps track, the FDA keeps track of this information. If this information amounts to anything, if there's a eye problem that shows up consistently or nausea that shows up consistently, there are measures that are taken to find out what's going on and monitor the drug. So that's the traditional drug discovery. What the government and the biotechs are doing are, first of all, the government has ordered millions of doses of the vaccine from AstraZeneca in the UK at risk. They ordered 3 million doses at risk. The vaccine hasn't even been provided yet, but they already have an order in for it. And in the case of Moderna, they're collaborating with the NIH. They actually have streamlined phase 1 to 45 patients, which is not unreasonable. But then they eliminated phase 2. Somebody arbitrarily picked a dose of 100 micrograms, and that's what they're using for their phase 3 trial. And their phase 3 is going to be run in 30,000 people. Remember that issue of safety, that inverse risk-benefit ratio. 30,000 people should be able to show that it's safe and effective. With 30,000 people to choose from, you can take African Americans, you can take Latinos, you can take any elderly, people with kidney conditions, people with liver conditions, anything you want, and fit them in as a statistically significant size group. The expense is immense. A clinical trial of that size would be probably unprecedented, but it could shrink the development time. So there's a variety of opinions in the field as to how or if this accelerated program of development for vaccine will produce effects. It's always a crapshoot, but the timeline in any event I think is going to be at least 2 years. That has a lot of implications for the social side of things and people with Parkinson's. We'll discuss that. Now, let's talk about the healthcare system inoculating at least 80% of the population of the United States. Why do I take that number? Because epidemiologists, people who study the pandemics and epidemics, have a way of calculating based on the virulence and transmissibility of a pathogen what portion of the population will have to be immune so that the virus, or in this case, can't infect enough people to make itself sustainable. It can't pass itself on to enough people to keep going. It's got to go back to the bat and wait there for the next jump across the species barrier into humans. So that's the first thing. Here it becomes immune because it's able to protect itself, because the virus just cannot infect enough people to keep itself rolling. So your infectivity has to be greater than 1. If you can only transmit the virus to one other person, you're never going to be able to increase the viral load in the population because you're only doing it one at a time, and eventually it's going to die out. And in other cases, such as the influenza virus, we have some years that you've got 30 or so % protection, and the other 70% are not responding. So vaccines have a whole variety of efficacies. It's not a question of it instantly being a neutralizing vaccine. My understanding is that the Moderna vaccine is neutralizing, and so is the Oxford vaccine. I hope that's true, and I hope that it holds up under the tests. But there is the dark side of this. And as I say, there is no AIDS vaccine. 33 years after trying. In this particular case, it's probably because the virus is hypermutable. It changes extremely quickly. COVID, or SARS-2, is not thought to be as mutable as the AIDS virus. But since we've taken such a quick step from the lab bench to the clinic, we don't know that much about the basic science of COVID or SARS-2. And we don't know as much about coronaviruses as we should know in order to be able to answer some critical questions, such as how hypermutable is it? So what have we learned? What takeaways do we have from this? The first is that we can exercise situational awareness to increase the reach of our immune systems by observing social distancing, which should be the norm if you really want to try to stay uninfected until a vaccine is raised and herd immunity is established. And my guess is that's going to be a good 2 to 3 years. So in many cases, it's not going to happen. People are just going to give up, start associating, and pay the price. There's two ways to induce herd immunity. One is to get the virus, survive it, and then be immune for the rest of your life. The second way is to be vaccinated. I'd rather do the second way. But again, this is a personal decision. This brings us to the second point, that you should make a conscious decision with your care partner on how you want to live out the process as it goes forward. Hope is not a strategy. You have to be active. You have to wear the masks and avoid crowds and wash your hands and do all the things that Tony Fauci and others advocate for public health before we have anything else. Because right now there isn't anything else and there's no vaccine. The other thing to do is keep your routine daily regimen of exercise. But if you go outside, make sure that you wear sunblock to prevent melanoma. People with Parkinson's are up to 7 or 8 times as susceptible to melanoma as age-matched controls. People who are the same age but don't have Parkinson's. If you don't go outside that much, make sure that you're taking vitamin D, because vitamin D requires sunlight to be activated in its normal metabolic form. You can't take it from Costco's vitamin D3. If you don't, you run the risk of osteoporosis. And osteoporosis is much more likely to occur in Parkinson's patients. It's better to light one candle than to curse the darkness. Thank you and be well.